All right. To remind you guys online, just because you're online, remember you still have to send in the attendance words. They're not extra credit for you. That's how you get credit. That's how I know you didn't just turn on your computer and roll around and go back to sleep, which would be very tempting to do on Friday, 8 in the morning. So yes, make sure you guys that are online send in those attendance words within 5 minutes of class. And speaking of attendance, let's get to it. Or they started. Let's see. Ryan's not here yet. Curry is not here yet. Darby is here. Fisher is not here yet. Hamilton, here. Hellman is here. Ashley is here in red, so we'll put extra credit there. Crystal is here. Taylor is here. Norris is here and red. Rollins is not here. Saunders is online. Shamblin here. Taylor Pierce, Vargas, and Young is online. And it's here. Did I miss anyone? All right, I'll do announcements before we get started. Hoping some more people show up. So I got some good questions before I start recording. Independent work. So by the end of this week, you should have 50 points of independent work. It's 10 points per week. And I reminded that person and somebody earlier this week. It's not like it's supposed to be 10 points. Yeah, it's supposed to hit 10 points per week, but it's not like one assignment is supposed to be 10 points and you turn in one assignment per week. Just keep turning stuff in until you hit the points you're looking for. I mean, if I were you, I would keep going until you hit 100. Because remember, right now, technically independent work is still worth, actually, I'm going to change it as of right now. It's been worth 14.5 points per page. So when I write you back and say full credit, that's what I've been meaning. But I'm going to drop it back down. Remember, it started at 10, and then I boosted it up to 15, and then I dropped it once. Right, so now I'm going to drop it again. Now we're down to 14 points per page, and I'm going to keep dropping it. So get writing. One of you is almost done. Actually, two of you are almost done with the whole 100 points. So yeah, turn stuff in. Another one of your classmates, I'm so glad they said this. They said, I've been writing it. I've been holding on to it. I was going to turn it in at the end. Don't do that. Because first of all, your grade is based on that 10 points per week. You know, so if you, especially if you're a student athlete and you're like checking your grade with me, then that's going to be based on how many of those points you have. So make sure you do that. Turn it in. Yeah, that's basically it. Independent work. Write the papers. Do all the other stuff if you want. If you don't want to write papers. Any questions about independent work? Okay. What else? Someone else asked me another question. What's it about? So you guys remember? Someone else asked me a question this morning. What's it about independent work? Yeah, can't remember. I haven't had all my coffee yet. Let me think before we move forward. Talking about wearing red. Talked about homecoming. So I guess that's it. Anyway, I guess maybe the other thing was also about independent work. If you want to know how many points you have, generally when you send something in and I grade it, first of all, I'll respond and be like, your new total is this many points. So that's one way you can find out. The other way is if you look on WVSU Online, which I admit is a little bit slow because I don't only update it every so often. But you can see it in increments of 10. So for example, if you have 22 points, if you look on WVSU Online, it'll say you have 20. If you have, 48 points, if you look online, it'll say 40. And I can explain why I do it that way if you want, but I'll do that some other time. So yeah, those are the different ways you can tell. And then of course you can just email me and ask me. But again, my suggestion, the best way to find out is submit something. And when I respond with the grade, I'll tell you how many points you have total. So any questions about independent work? All right, let's jump back into this then. To remind you, we just finished the overview of cellular respiration. So what we're going to do now is go into the details. We're going to talk about glycolysis and citric acid cycle and electron transport chain. But before I do, I'm going to do a brief review and make sure you guys understand all this stuff. Because if you don't know that, which I don't expect you guys to have it memorized yet, but I want to make sure you understand this before we start talking about it in detail. So let me ask you this. In which of the 3 stages is ATP made? Does anybody know the answer to that? I think you said All of them, yeah. Whoever said that is correct. All three. Of course, the most are made in electron transport chain, which is very important. You definitely need to know that, right? The electron transport chain is where the most are made. How about carbon dioxide? I remember which one that's made out of, or which one is producing that. Carbon dioxide is the hint. The way I say carbon dioxide with a C. No guesses? So like I told you, in this exam, anything with carbon, whether it's being used in photosynthesis or produced in respiration, it's going to be a cycle. In this case, the citric acid cycle with the carbon, C. And which of the three stages is water produced? And I'll give you a hint. It's produced when the oxygen accepts some electrons. Good. Electron transport chain. Slash phosphorylation. How about glucose? Which one is glucose used, or which one uses glucose? And that's kind of in the name too. It sounds a lot like glucose. Good, glycolysis. And then finally, and which one is oxygen used? And I already basically gave that answer away earlier when I was talking about water, because oxygen and water kind of go hand in hand in these conversations. The ETC. All right, now that being said, are almost ready to move forward. Of all these things on the board, which of these is the most important? Like, what are we after? Like, what is the purpose of cellular respiration? Is it like the purpose of it to produce carbon dioxide? Is the purpose of it to produce water? Is the purpose to use glucose? Is the purpose to produce oxygen? Like, what are we really after? What are your cells really after when they're going through this process? Of all those things, what are they really after? What are they either trying to produce or use? One of those is the most important. The other is just part of the story. And you should really know this. I'm glad I'm asking this, because if you don't know this, there's no need to talk about the rest of it, because this is big picture stuff here. If you don't know the purpose of cellular respiration, then who cares what stage this is done and that is done? Like, what is the purpose? What are we after? And I remember saying this in the last chapter. I said, if I could boil this last chapter down to the one sentence, this would be it. And I mentioned one of these things in that sentence. Come on, think back. And if you're really good at digging into details, you can look at the board and kind of see the answer in a way, depending on how your brain works. All right, what did we talk about last chapter? Or, yeah, what did we talk about in the last chapter regarding letting things in and out of the cell? If something can't diffuse naturally into a cell, does anybody remember how we get it into a cell? Glad to stop. Just a good positive like that. Active transport, right? This thing is diffused from an area of high concentration to low concentration. But if in your cell you already have a high concentration of whatever it is your cells need, and a low concentration on the outside, it's not going to diffuse in, right? Because that's not how diffusion works. So we need active transport. We need energy to pump things in. Of course, we need energy for a lot more than that, but this is one of the things we talked about in the last chapter. So if we need energy, where does that energy come from? Directly, not ultimately. Ultimately, it comes from the sun. Directly, where are your cells getting that energy to do that? Earlier in this chapter, we talked about enzymes, because we're talking about chemical reactions when we're building stuff, right? Like, I don't know, X plus Y gives us XY. Your body's trying to build, or your cells are trying to build a molecule. That takes energy. Where are they getting the energy from? Yeah, enzymes lower the activation energy to do that. But we talked about all this. What amongst this do your cells need? What are we after for cellular respiration? Energy. High energy molecules. The only one of these molecules that I've described the shape and the function, the structure. I talked about how that structure was so energetic and why it was so energetic. Nothing? Are you guys just being, like, wrong answers are fine. Are you just someone afraid to give the wrong answer? ATP. Yay, thank you. Good. Like I said, from the last chapter, if you forget everything from the last chapter, your cells need ATP to do work. And by work, I don't mean like making a living so they can pay the bills. In the cellular sense, doing work means being alive, right? We need to be able to, your cells need to do work. They need to do things so we can be alive. All right. Finally, that being said, this whole thing is all about making ATP. Now, this is a little bit less important, but still pretty important. Of the three stages, in order, there's glycolysis, citric acid cycle, and electron transport chain. Of those three stages, which one produces the most ATP? Since that's the most important thing anyway, we need to focus on that. What's producing the most ATP? Because all three are producing ATP. Which one's producing the most? And I feel a little bit less bad that you guys don't know this because I emphasize the ATP thing a lot. But all right, so you guys don't know this one? Electron transport chain. It's all about the electron transport chain, electron transport chain, electron transport chain. Make sure you know that. Not only is that going to be one test question, which are the three stages producing the most ATP, but to truly understand this whole process, you need to know that this is all about the electron transport chain. It's all about that third stage. So that being said, before we move forward, keep this in the back of your mind. This is the story of cellular respiration. We're after ATP. That's the main purpose here. Electron transport chain is the one that's doing most of the work. And it is doing that because it's using high energy electrons, right? Electron transport, this is stuff I've already told you with the whole big picture thing. Electron transport chain uses high energy electrons to produce a bunch of ATP. So that being said, the other two, glycolysis and citric acid cycle, yeah, they produce a little bit of ATP. But the big picture here is that they're all about stripping away high energy electrons. It's that third one that needs electrons. Make sense? All right, keep that in the back of your mind as we go through this in detail. Here we go. During glycolysis, we have a 6-carbon glucose that is split in half to form 2 molecules of pyruvate. Each one has 3 carbons. Circle what you need to know from that bullet point. We start with glycolysis. We end with pyruvate. The exam, that's it. Yes, technically we don't start with glycolysis. That's the process. Sorry. I can't erase it. We start with glucose, we end with pyruvate. You need to know that for the exam. And that'll be 2 test questions. Which of the 3 stages uses glucose? Glycolysis. Which of the 3 stages produces pyruvate? Glycolysis. Yes, technically that that glucose is 6 carbons, C6H12O6. And yes, technically when you cut that in half, you're going to have 2 3-carbon molecules, but you don't even know that. All you need to know is you start with glucose and then with pyruvate. You guys with me so far? All right. Now, when we break down that glucose, when we break it in half, the purpose, remember, big picture here, we're trying to get high energy electrons. So by breaking that in half, this is what this is, right? Those high energy electrons are stripped away. NADH is taking them to the electron transport chain. And to remind you, these things are kind of basically the same molecule, NAD plus and NADH. This is just technically the name before it gets the electron, right? It has the plus, meaning it's ready to accept the electron. And then once it gets the electron, it's technically called NADH. So yeah, again, start with glucose, end with pyruvate. The whole purpose of this process is to produce or to harvest those high energy electrons. So that's what we do there. Another thing you need to know, and I've already mentioned this a couple of minutes ago, is yes, we do produce a little bit of ATP. The net gain is technically 2. I don't care about that. So remember, the 2 test questions you're going to have regarding what makes ATP, one of them is going to say, which of the 3 stages make ATP? The answer is all 3. And then the other one is going to say, which one makes the most ATP? And the answer is electron transport chain. So the only thing you need to know about glycolysis as far as ATP is concerned is, yes, a little bit of is produced. More importantly, for the big picture, we're stripping away high energy electrons. So then later, the ETC can make a bunch of ATP. Now, I'm going to put a big old X to this because your book talks about it, so do I. But I'm not going to ask you about it. But yes, glycolysis itself is broken down into 2 stages. And I don't even think your book names them. I don't remember. But I think they mentioned the fact that there's 2 stages, but I don't think they gave them the names. They mentioned the names, but technically it's energy investment, energy harvest. I'm going to show you pictures of how this works. But again, you don't need to know that. But yeah, for attendance, let's say the first word for attendance is half, right there. Half. Like a lot of us seem to be half awake. I know I'm half awake. All right, any questions about this slide before we see some visualization of the words that are on this slide? All right, I kept the pictures from the old textbook because you guys have your own pictures from the old textbook. I wanted to show you some other ones that maybe one of them clicks with you a little bit better. Another good test question based on this picture is, which of the three stages happens outside of the mitochondria? The answer is glycolysis. Yeah, but again, in glycolysis, we break down, of course, I don't visualize this, but we break down glucose to form pyruvate. In the process, we form a little bit of ATP. More importantly, we're stripping away high energy electrons that are eventually going to go to the electron transport chain. That's where the meat and potatoes is at. That's what's important. So again, the input is a 6-carbon glucose. Not that you need to memorize the fact that it's 6 carbon. That is broken in half to give us 2 3-carbon molecules that are called pyruvate. And again, I don't care that you know that they're 3 carbon, and I don't care that you know that there's 2 of them. The point is, we go from glucose to pyruvate. You need to know that. Which of the 3 stages uses glucose? Glycolysis. Which of the 3 stages uses pyruvate? Glycolysis. Any questions about this picture? All right, like I said before, technically this is broken down into 2 stages. You don't need to know this, but I'm going to talk about it very quickly because your book did. So you need to, like I said earlier when we talked about enzymes and chemical reactions, even chemical reactions that put off energy, like this one, right? Because we're gaining energy here. That still requires a little bit of input of energy. So we do put 2 ATP in to begin with. And then, yeah, the more important thing here, and again, you don't need to, come on, you don't need to visualize it like this, but that's what we're after right there. We get stripped away high energy electrons. And yes, technically, this pyruvate has 2 phosphate groups, but who cares about all that? Anyway, those two pyruvates are then stripped away a little bit more. We take away some of the phosphate groups and make ATP. But again, this picture, if you're studying, I wouldn't even study this picture. I just included it because your book did, so I wanted to talk about it. That way, when you read through your textbook and you see this, you're not like, so did you forget to talk about this? No, I'm talking about it, and I'm telling you, you don't need to worry about it. Any questions about this? All right, so again, not that you need to know this, but I'm going to address it. This 3-carbon molecule has 2 phosphate groups, and that's where the ATP comes from, because remember I did say glycolysis does produce a little bit of ATP, and that's where it comes from, because there's an enzyme that takes off the phosphate group and adds it to the ATP. But again, you don't need to know this. I'm just including it because your textbook did, and I don't want you to think I forgot it. So any questions about glycolysis? We're not done talking about glycolysis. Excuse me. All right, let's talk about citric acid cycle next. It's interesting. Your textbook puts these 2 things together. Citric acid cycle and, as they put it, oxidative phosphorylation. I always have problems saying that. AKA the electron transport chain is what that second one is. But they put it together. Let's talk about it. But learning objectives, according to your book, by the end of this section, you'll be able to describe the location of the citric acid cycle and oxidative phosphorylation in the cell. And I've already told you that because remember I just said which of the 3 stages happens outside the mitochondria, and the answer was glycolysis. Therefore, by process of elimination, these other 2 happen inside the mitochondria. Describe the overall outcome of the citric acid cycle and oxidative phosphorylation, aka the electron transport chain, in the terms of the products of each. Describe the relationship of glycolysis, the citric acid cycle, and oxidative phosphorylation, aka electron transport chain, in terms of their inputs and outputs. So here we go. Here's the thing. Actually, it's a good time to see if anybody's awake. What is it that we ended up with in glycolysis? We just got done talking about glycolysis. We started with glucose, got a little bit of ATP, stripped away some high energy electrons, which was the important part. But then what were we left with? What happened? What was that molecule that we made when we broke that glucose in half? Pyruvate. Thank you. Good. So obviously that's where this story starts. Pyruvate. Now, remember, like I said, glycolysis is happening outside of the mitochondria. So that pyruvate needs to get inside the mitochondria for this to happen, right? Problem is, they can't cross the border, so to speak. So, as you know from the last chapter, some things can come in and out of the cell membrane, or in this case, the mitochondrial membrane, willy-nilly. Some things need help. and this thing needs help for whatever reason, and you can look it up for independent work if you want. For whatever reason, pyruvate cannot enter the mitochondria. Therefore, it has to be converted or groomed. You want to think about it like that. So let's talk about what happens there. If you're taking notes, there's no need to write any of this down yet. I'm going to explain the whole thing, and then once I've explained it, then I'll tell you what you need to know. So when we groom this pyruvate, we do some things to it. And one of the things we do is we knock off a carbon. Because remember, like I said, glucose is 6, 2, 3, 4, 5, 6 was glucose. It was 6 carbon molecules, and that gave us 2 3 carbon molecules, which are the pyruvate that we're talking about, right? So those are 3 carbons. But now we're knocking one off, right? We're getting rid of one of these carbons, one each. And that's where we're getting the carbon dioxide, which is a test question. Yeah, I guess if you're writing that down, you can write that down again. I said it before. Which of the 3 stages produces carbon dioxide? Citric acid cycle. And that's one of the reasons why. But you don't even need to know that. I'm telling you where it comes from. But there's not going to be a test question that says, in the citric acid cycle, where does the carbon dioxide come from? And if that was a question, then the answer would be, well, that's when we took a carbon off of the pyruvate. But you don't need to know that. Moving forward, technically, once you lob off that carbon, then it's no longer pyruvate, right? As you can imagine, if you go from a 3-carbon thing to a 2-carbon thing, even though you're not chemistry majors, you can just figure, obviously, that's a different molecule. It's going to have a different name. And it is. It's called acetic acid. So know that. Actually, go ahead and write that down. This is one of those few things that's not in your textbook that I'm going to require you to know. There will be a test question that says, which of the 3 stages is associated with acetic acid? And here's a little hint, because it won't just be acetic acid. Oh, yeah, I guess it will. The way to remember that is acid. Which of the 3 stages is associated with acetic acid? I know it's a different type of acid, but it's the citric acid cycle. So acids will always go together. Citric acid cycle is associated with acetic acid. And obviously also citric acid, but yeah. And I'll think about that. I might make that one an extra credit question since it's not in your textbook. But I am telling you that's going to be on the exam. It's already on the study guide. Which of the 3 stages is associated with acetic acid and citric acid cycle? Any questions so far? All right. More importantly, like I said, yeah, we produce some carbon dioxide. That's just a byproduct. So that's not important. Technically, yeah, we've made acetic acid. That's not important for the grand scheme of all cellular respiration. So more importantly, in this process, we've stripped away high energy electrons. But you already knew that, right? I already told you the first 2 stages are just stripping away high energy electrons. That's the big picture. That's what the first 2 stages are for, really. All this other stuff is basically like the side story, right? It's not even that important. What's important is we're stripping away high energy electrons. And again, to remind you, anywhere on this exam, when you see a molecule that looks like this, the 2 other ones that I'm going to show you, one of which I'm going to show you soon, but if it looks like that, you should think, oh yeah, that's that thing that's moving high energy electrons. Any questions so far? All right. So this acetic acid, this 2-carbon acetic acid, it still cannot get into the mitochondria. And again, what makes sense to you why it can't make it in there. But what you can do is look it up for independent work. Why, first of all, again, why can't pyruvate go into the mitochondria? Why can't acetic acid go into the mitochondria? All right. So because of that, each one of these 2 acetic acid enzymes is then attached to a molecule called coenzyme A to form something called acetyl-CoA. You don't need to know all those details. If anything, I ask you this. Which of the 3 stages is associated with acetyl-CoA, which is coenzyme A? And the answer is citric acid cycle. So if anything, you just need to know that acetyl-CoA is associated with citric acid cycle. So again, I've got a lot of details here, but to remind you, you don't need to remember that pyruvate lost a carbon to carbon dioxide, and that left us with acetic acid, and that had to be attached to something called acetic, or to coenzyme A, and that gives us acetyl-CoA. You don't need to memorize those details, but you do need to memorize that acetic acid is associated with citric acid cycle, and acetyl-CoA is associated with citric acid cycle. And I think one of the only things that would be tricky about this, that I'm thinking about it, Pyruvate, right? Technically, pyruvate could be mentioned twice in the exam, depending on the question is worded. Which of the two stages starts with pyruvate? Or excuse me, which of the three stages starts with pyruvate? Then the answer is citric acid cycle. Which of the three stages ends with pyruvate? That'd be the one we just talked about, right? Glycolysis. The big picture stuff. Anyway, once it's attached to the acetyl-CoA, it is then ready to go. It is ready now to go into the mitochondria and get into the citric acid cycle. So technically, we haven't really been talking about the citric acid cycle yet. This is all about getting ready for the citric acid cycle. But as far as I'm concerned and your textbook is concerned, we just lump this all into citric acid cycle. Anyway, any questions about this slide? All right, again, another picture, not from your textbook, because the one from your textbook tells the same picture, but it tells the same story, but in a different way. So to remind you again, the two pyruvate molecules for glycolysis with room, right? We lopped off some carbons, make carbon dioxide, we left with acetic acid. That was attached to coenzyme A, which gave us acetyl-CoA. That is then able to enter the mitochondria, which is where the citric acid cycle happens. And in the process, again, we're stripping away high-energy electrons while making ATP. We still have more to talk about with citric acid cycle. Yeah, this picture is not from your textbook. This is everything I just said, but again, it's just visualized. We started with this pyruvic acid. We lobbed off a carbon to make carbon dioxide. We were left with acetic acid. We're stripping away high-energy electrons. This is the important thing. We're attaching it to the coenzyme A, which gives us acetyl-CoA, and then it's now ready to go into the citric acid cycle, which is what we're about to talk about, or what we're about to talk about. All right, so it makes it into the mitochondria. It loses its coenzyme, so now we're back to acetic acid. Not that you need to know that detail, because remember, the only thing you really need to know, if anything, is if I say which of the three stages is associated with acetic acid, the answer is citric acid cycle. At this point, two molecules exit as a waste product, two molecules of carbon dioxide. So again, just like the last one I talked about, which is still citric acid cycle, with that grooming process, we produce carbon dioxide. That'll be the test question. Which of the three stages is associated or produces carbon dioxide? And the answer is citric acid cycle. Like I said earlier, yes, we're making a little bit of ATP. So again, which of the three stages produces ATP? All three. And again, much more importantly, we're stripping away high energy electrons. And those are the molecules that are doing it. And there's the other one I told you about. See, look, FADH2 looks a lot like NADH, right? Those things look similar in writing. And they are similar in structure, and obviously almost identical in function. So again, in this exam, when you see anything that looks like that, and the third one that I'm going to introduce you to for photosynthesis, you should think, yep, that's the thing that's carrying the electrons. If you want for independent work, you can look this up. What are the differences between those two? What's the different? First of all, what's their actual names? I think that's actually in your textbook. Then you could look up what's the different structure? If they have a different structure, do they have a different function? Like, why can't FADH2 do the same thing that NADH do? Might be a little bit over your head. You can look it up if you want. The next word for attendance will be recycled. Anyway, all the carbon atoms that enter the citric acid cycle as fuel are accounted for as carbon dioxide in a 4-carbon acceptor molecule, which is recycled. So that bullet point, as far as the exam is concerned, is useless. I'm not going to go into that much detail, other than the fact that, like I've already said, carbon dioxide is produced in the citric acid cycle, and you do need to know that. Questions about these words that are on the screen before I show you a picture that illustrates all these words? Picture, again, not from your textbook, because your textbook has a picture. This tells the same story, just in a different way. So again, the inputs, not that I'm going to ask you the inputs and outputs necessarily, but you start, the acetic acid goes in, we need some ADP plus some phosphates, we need some acceptor molecules to get this to the high energy electrons. But here's something interesting. Maybe I'm thinking of the other one. Nah, that's good. I don't even talk about it yet. So here we go. We have a 4-carbon acceptor molecule that I don't even talk about, but it has a name that we don't even talk about. It gets attached to that 2-carbon acetic acid molecule, which gives us citric acid, which is a 6-carbon molecule. If that thing gets broken down, we lob off 2 more carbons, right? Because 6 minus what? It's 4. 6 minus 2, right? So we lose 2 more carbon dioxides. We make ATP. Most importantly, we're stripping away high energy electrons. So again, test questions on this. Which of the 3 stages is associated with acetic acid, citric acid cycle? Which of the 3 stages produces ATP? All 3. Which of the 3 stages produces carbon dioxide? Citric acid cycle. Which of the 3 stages is harvesting high energy electrons? Glycolysis and citric acid cycle. That's it. That's it for citric acid cycle as far as we're concerned. I think it's a lot more complicated than not in your textbook and not in this course. If you have any questions about the citric acid cycle or glycolysis for that matter? So think about it. When we talked about glycolysis, we started with glucose and ended with pyruvate. Here, we don't necessarily start or end with anything, right? Because, I mean, technically, yeah, we start with this acetic acid, and then that goes into the cycle, and the cycle just continues, right? So we don't really start and end with anything, so there will be no test questions like that. So now when we talk about this 3rd stage, we're not really talking about starting and ending materials either. But we are talking about this. What have we been harvesting this whole time? High energy electrons. So that's the story we're about to talk about. In cellular respiration, these electrons are falling in a cascade down an energy staircase. Because notice I keep saying high energy electrons. I don't just say electrons, I say high energy. We don't have enough time to teach what that means by high energy. Just think they have a lot of energy. They're not just regular electrons, they have a lot of energy. But as they go through cellular respiration, they're basically kind of just visualize it as if they're falling. Like a ball falling down a staircase. When it's at the top of the staircase, it has a lot of potential energy, right? And it's falling and it has kinetic energy. The lower and lower it gets, the less potential energy it gets. It's releasing that potential energy, basically converting it to kinetic energy. But the same thing here. You can visualize that we start off with the electrons, high energy place. Glucose. Glucose is a high energy molecule. Glucose. There we go. Energy. Energy right there. Anyway, we start there, right? And as it goes down, these electrons are losing energy. They're releasing it. Because remember, energy can't be created or destroyed. So I guess I shouldn't say losing it. Those molecules are losing it, but the system itself keeps the energy, it converts it. Now, how many of you have ever jumped a dead battery, like a car battery or anything, or seen that, right? So when you do that, there's 4 of these things, right? Which ones need to be attached to something? Or you need to be able to jump the cable or jump the battery. Can you just hook up one cable? How about 2? 3? Well, you need all 4 though, right? You need all 4. Because, and I don't know how much you guys know about electrons or electronics. That's what I used to do in the Marines. Electrons have to come from somewhere and they have to go from somewhere. Right? So all the electrons that are flowing this computer came from John Amos, and they're technically going to the ground. So they came from somewhere, they're going somewhere. And the same is true here. This is basically an electric circuit. The electrons have to come somewhere, from somewhere, and they have to go to somewhere. And that will also be a test question. Where do these high energy electrons come from? The answer is this: glucose. High energy electrons are coming from glucose. Yes, technically we should also strip them away from pyruvate. We technically strip them away from the acetic acid. And we technically strip them away from the citric acid, but that is all a byproduct of glucose. So the answer to that question is, where do these high energy electrons come from? Ultimately, they came from glucose. Which will be very important in the next semester, or next chapter. This next chapter, we're going to talk about how we build glucose. Part of that story is we've got to pump in some high energy electrons. But anyway, yes, keep this in the back of your mind as we move forward. Electrons have to come from somewhere. They have to go somewhere. I'm about to tell you, but this, and I've said it before, I'm not going to say it again yet, but off to see if anybody remembers. Where do the electrons go? They started at glucose. Does anybody remember where they end up? That's okay. I've told you before, but it's okay that you don't know it yet. By the end of this, you do need to know it. Memorize it. Anyway, just to remind you, this is also happening in the mitochondria. And actually, we're going to talk about why that's important. For the first time, that's actually relevant. The fact that glycolysis happens outside the mitochondria, not that important. The fact that the citric acid cycle happens inside the mitochondria, not that important. But the Mitochondria itself is part of the story for the electron transport chain, as you're about to see. So again, like I said, the overall effect of the electron transport is the downward trip for electrons. And by downward, I mean, again, we start at high energy, we end at low energy, just like a ball going down the stairs. Again, like I said, these high energy electrons come from glucose. And then they are stripped away by NADH and also technically FADH2. Not that I'm going to get that specific. When you see in molecules like that, again, you should think of something moving electrons. And again, like I said, here's the answer to that question I just asked you. They come from the glucose and they end with the oxygen. And you need to know that. Electrons start in respiration. Where do the electrons come from? Glucose. Where do they end up? Oxygen. Oxygen is the final electron acceptor. And when that happens too, then that forms a molecule of water, which again is another test question. There's a lot of test questions on here. Which of the three stages uses high energy electrons? That would be electron transport chain. Because remember, the other test question was which of the three stages strips away high energy electrons? And the answer is glycolysis and citric acid cycle. Which of the three stages uses oxygen? The answer is electron transport chain. Which of the three stages produces water? And the answer is electron transport chain. Again, like I said, Wednesday, these two words should always go together. Whether we're talking about photosynthesis or respiration, those things are always related. Oxygen and water are always related. Here, oxygen is catching electrons, so to speak, and that forms water. And then it's completely opposite for photosynthesis. Water is like, hey, get some regular old electrons so that they can be energized. And that forms oxygen with photosynthesis. Any questions about this side? All right, here's a visualization of everything I just said. Again, a picture not from your textbook. I wish your textbook did have this picture, but here we go. So again, we have electrons from food, or we're narrowing it down to glucose. Like I said when we started this conversation, I'm going to be talking about glucose, but technically everything we eat, we break down. So yes, it's other stuff too. Anyway, we strip away these high energy electrons. That's why they're up here, because they're high energy. As they go down the electron transport chain, they're releasing energy. We're using that energy to produce a bunch of ATP. We're going to talk about how this works here in a second. Then those low energy electrons, if you want to call them that, they're accepted by the oxygen, and that produces a molecule of water. And notice your book is really specific. Technically, yes, this NADH is carrying 2 electrons, right? So then technically, 2 electrons is meeting up with 1/2 a molecule of O2, which then meets up with 2 molecules or 2 H+ ions, but you don't need to know all those details. Again, where do the electrons come from? Glucose. Where do they end up? Oxygen. And then when oxygen does that, then we form a molecule of water. The rest of those details you don't need to worry about. Which works out. It's not your textbook. Any questions about this before we talk about this? So all this shows is that these electrons are putting off energy as they go down their steps, so to speak. And that energy is used to make ATP, but this doesn't really explain how. So let's talk about how that works. So there's proteins of the electron transport chain / oxidative phosphorylation. And they're built into the inner membranes of the mitochondria. There's going to be a really good picture. Actually, yeah, just wait. I'll just show you the picture instead of me drawing it. These membranes are highly folded. I'm not going to ask you that, but that'll make sense. I'm going to tell you why they're highly folded when I show you the picture. Well, I'll tell you now, but it'll make more sense when I show you the picture. They're highly folded because it gives you a large surface area. So obviously, I guess I will draw all this. That's the outer membrane, the inner membrane. And like I said, like this says right here, you got the protein built into the membranes. Obviously, the more membrane you have, the more proteins you have, right? If it's those proteins that are making ATP, the more the merrier. So that's why we want a large surface area. But again, I'm not going to ask you that on the exam. So let's look at a picture of what I'm talking about. The drawing to me is better than the actual electron scanning microscope. But yeah, this is what it looks like. So there's the outer membrane, and this is like we cut away the outer membrane so we can see the inner membrane. And then also we cut away the inner membrane so we can see the inside. But the point here is we have this gap. Actually, I guess I will draw it because that picture's not good for what I need. Again, we have the outer membrane, we have the inner membrane. So here we have this little area in between. That's going to be an important area. You'll see why here in a second. There's this little area in between. That will be important. Anyway, that's just a picture. You don't need to know any of these, wordings. I'm not going to ask you the parts of the mitochondria, but that's what it looks like. That's what it's called. But yeah, the space between the membranes is going to be very important, and you'll see why here in a second. But again, imagine, I'm going to try to draw this. Imagine these are all proteins built in to the membrane. I don't know if you guys can see those little red dots. So that's why it's so folded, right? Again, if the proteins are embedded into the membrane of the inner membrane, then the more you have, the better, right? So anyway, here we go. The energy stored by the electron transport is something like water behind a dam. So I'm sure you guys probably know how a dam works, even if it's not hydroelectricity technically. If you can imagine, here we have like a little stream of water. But try to picture in your mind what it looks like on the other side, right? This huge lake or big body of water anyway, right? And that water wants to come down. Gravity wants to take it down. That's what it naturally wants to do. And the same thing kind of happens here in the mitochondria. What we have is a bunch of H plus ions, a bunch of them in between the membranes, a bunch of H plus ions, just like we have water behind a dam. And just like gravity wants to take that water down hill, right? In this case, what we have is diffusion. We barely have any H plus ions in here. So diffusion wants to make them go from an area of high concentration to an area of low concentration. Does that make sense? This is finally why diffusion was important. Because otherwise, the rest of the semester, it doesn't matter about diffusion. That's why you have to learn diffusion. This is why it's important. These hydrogen ions want to diffuse down the concentration gradient. And in a sense, it is like water behind a dam. And just like in some dams, you could attach a turbine to it and produce electricity. So the water runs down, it just flows downhill naturally, right? Turns the turbine, produces electricity. Same here. These hydrogen ions want to come down, and they do. And your cells have their own versions of turbines. And they literally do spin. And they are called, you need to know this, ATP synthase is the enzyme. That is the enzyme that is making the ATP in the electron transport chain. Should be easy to remember because it's in the name. Like I said, all enzymes, for the most part, as far as we're concerned, end with ACE. And they're either named after what they do or named after the substrate. And in this case, they're named after what they do. ATP synthase. They synthesize ATP. So yes, you can consider them like little turbines in a hydroelectric plant. These hydrogen ions are flowing downhill. They're getting diffused, so to speak. They are going through diffusion, going through those ATP synthases, and it literally does turn a turbine, so to speak, and that's how we make the ATP. So a good test question. Which of the three stages uses ATP synthase? The answer is electron transport chain. Yeah, that's it for test questions on that side. Anyway. Any questions about that side? Here's another picture. Your textbook doesn't have a good picture of it. I think this is a great picture. So again, you've stripped away these high energy electrons, and they are delivered to the electron transport chain, and they literally go from, just like any other electrons in an electrical circuit, they go from thing to thing, right? So here we're going from protein to protein to protein, and they're giving off energy, and that energy is used to pump hydrogen ions against the concentration gradient, right? So again, this is why you have to learn about facilitated diffusion, or excuse me, active transport, because you need to understand this concept. There's a lot of hydrogen ions out here, or in between the two membranes. And there's not many hydrogen ions here in the inner part of the mitochondria. So it's going to take some energy to pump these things against their concentration gradient. And that's where the electrons come in, right? They give them the energy to pump them against their concentration gradient. But again, they are against the concentration gradient. So now, again, we have high concentration, low concentration. We know that nature is going to want to do its thing. They're going to want to diffuse down. For whatever reason, they can't go. Luckily for us, they cannot go directly through the membrane. So they have to go through the ATP synthase. As they do, that energy is used to produce ATP. So there's a lot of test questions on here that I've said before, and I'll keep saying it again. Which of the three stages uses high energy electrons carried by NADH? Electron transfer chain. Which of the three stages, oh, here's one I haven't said yet as a test question. So you write it down if you need to. Which of the three stages pumps hydrogen ions against the concentration gradient? Electron transport chain. Which of the three stages, or in which of the three stages, do hydrogen ions flow down the concentration gradient to produce ATP? Electron transport chain. Or a little hint for you, if any question asks anything about hydrogen ions, the answer is electron transport chain. Whether we're talking about pumping them against the concentration gradient or letting them come down the concentration gradient, the answer is electron transport. Which of the three stages produces the most ATP? Electron transport chain. Which of the three stages uses water? Electron transport chain. Which of the three stages produces, let me back up to the last thing I just said. Which of the three stages uses oxygen? Electron transport chain. Which of the three stages produces water? Electron transport chain. Yeah, so there's a bunch of test questions on that side. And you'll notice there's a little skull and crossbones right here. You might want to venture a guess what that means. It's because Captain Blackbeard with a microscope was the one to discover this third protein. It's because if you don't, that's the part that kills you. Like when you die, a lot of times that's what you're dead from. If you don't get oxygen, if you drown or choke, that's what kills you. There's no oxygen, therefore the electrons don't have anywhere to flow. Therefore, none of that happens. It would be like trying to jump those dead batteries, but you didn't put one of those things, you didn't connect one of those things. That makes sense. Like the electrons aren't going to flow unless you connect them. Same here. If you don't have oxygen, the electrons aren't going to flow. These things aren't going to be pumped against the hydrogen, against the concentration gradient. Therefore, there's nothing to flow down the concentration gradient. Therefore, there's no ATP. Therefore, you die. Your cells can't do the job that they need to do to stay alive. Anyway. Any questions about this slide? That'd be good timing. The next word for attendance. Let's do dinosaur for the next word for attendance. Anyway, another word, another number you need to know, and I've told you this previously in other discussions. It's about 32 ATP produced per molecule of glucose. That number will differ depending on your source. If I remember correctly, your textbook even refuses to give an exact number for that reason, because it differs. But for test purposes, you need to know about 32. And again, I'm not going to be tricky, so your multiple choices aren't going to be about 32, about 34, about 36, or about 38. It's not going to be like that. It's going to be in the tens, right? So 32, 52, 82, something like that. So any questions about that? Pretty simple. It's just a number you need to know, about 32 ATP per molecule of glucose. So I didn't make this mean. I don't make any of them. But that's a good independent work topic if you want to think about it. If cellular respiration requires ATP, because remember the first stage of glycolysis, which is the first stage of cellular respiration, I told you that it requires a little bit of ATP to keep this whole thing up. So if cellular respiration requires ATP, at the very first step, and cellular respiration makes ATP, right? That's where we get all of our ATP. Then where did the first ATP come from? That is a good question, right? The process that makes ATP requires ATP to get started. Sort of like that question, what came first, the chicken or the egg? Because that's an easy answer, the egg came first, but we'll talk about that later. So here we go. This is a big overall picture of what we've talked about. We're talking about, we're focusing on the big picture here. So again, these first two stages are all about stripping away these high energy electrons, which are taken to the electron transport chain, where those high energy electrons give off their energy, right? So they can pump hydrogen ions against the concentration gradient, so they can diffuse back down, and we can make a bunch of ATP. So again, this third stage makes the most ATP. The rest make ATP as well. But the big picture is they're trying to strip away the high energy electrons. Glycolysis is the only one that happens outside the mitochondria. Big picture stuff. Any questions about that picture? And that's, again, it's not from your test, from your textbook. So while you're studying, if you look at your textbook and you don't like the picture, remember, you've got a PowerPoint to look at too. It's the weekend, almost. So we wait, how many? I think it's only one of you over 21. Oh, a couple. Okay, so this is relevant, maybe. It's homecoming weekend. Let's talk about fermentation really quickly. Learning objectives. Even though we're not going to be able to finish this section today, but let's at least start it. By the end of this section, you'll be able to discuss the fundamental difference between anaerobic cellular respiration and fermentation. And you should be able to describe the type of fermentation that readily occurs in animal cells and the conditions that initiate that fermentation. So, cells can work a short period without oxygen. That's why, like, how many of you ever held your breath before if you're swimming or something? Did you die? Good, right? None too. I didn't die because, yes, for a short period, humans can go without oxygen, and that's what fermentation is. Fermentation, you need to maybe know the definition. Probably not. I'll tell you what you need to know later. But the definition is anaerobic harvest of food energy. So this fermentation. That's the book definition. I think a better definition is the production of ATP without oxygen. So the last word for attendance would be Taylor Swift. You guys might be too young to remember, but that's when he was interrupting Taylor Swift. So give her the honor there, not him. And that's it. I will see you guys on Monday unless you need me for office hours. Well,